Good morning, good evening, depending on wherever you're logging into this talk from. From BITSA, it's a great pleasure to have you all on this talk. I'm Srikar Nageli from 2007 batch, 2007 A5PS606P. And it's a great honor to be inviting the speaker for today's talk. So we have with us Professor S.S. Vasan from 91 batch. He did his bachelor's in chemistry and chemical engineering dual degree. He went on to do his master's from IISc and then also his PhD from Oxford and MBA from universities in UK. So he's currently heading the Dangerous Pathogens Team at CSIRO, and he has been in the forefront of research related to different pathogens or viruses that have been affecting the humankind. And most recently, the news that broke out about the researcher that was able to isolate the large quantities of the virus outside of China. So the credit goes to Professor Vasan, and he has made the entire community of BITSians all over the world proud. And so the talk that we have today is titled Understanding the SARS-CoV-2 virus for COVID-19 vaccines. So all the people who have questions about why is it so challenging to have the vaccine and things like that, this is the talk for you. So over to you, Professor Vasan. We are very excited to have you and looking forward to your talk.
Thank you. Thank you very much. Can you hear me well?
Yes, we can hear you.
Okay. So the title of my talk is first to understand SARS-CoV-2, and we can then talk about the vaccine landscape. I want to start by acknowledging the traditional owners of the land I'm joining you from, and that's the Wadurung community. I would like to pay my respect to their elders, past, present, and emerging. I always like to highlight one of the elders, and the one I picked today is Stan Dryden, also called Yara Munwa. Check him out. He's a great artist and a musician and a businessman. First of all, let's try and summarize where we are at the moment in terms of COVID-19. The number of confirmed infections are nearing 20 million, over 700,000 deaths around the world. So to put that in context, that's 1 in 27 people who have a confirmed infection have died. United States has about 25% of the burden, so just over 5 million cases. It's striking to see that the next 4 countries are what we call the BRICS countries, except China. So Brazil, India, Russia, South Africa. So the virus has actually had quite a telling impact on the emerging economies. So I also wanted to highlight the cases in the UK as well, because the worrisome aspect of the UK is with an aging population, you have substantially higher deaths. And I've also highlighted the Indian subcontinent, which are not far behind in terms of number of cases. So Pakistan and Bangladesh. The death rate in India and Pakistan is quite, in the subcontinent, is quite similar, about 1 in 50. The American death rate is quite similar to the worldwide average. So mainland China has had much higher deaths, but note that they are actually currently number 30 in the case burden. The second thing I would like to say is put this whole talk in context. number of cases, if you look at the confirmed number of cases, so about every 1 in 250 odd cases, to be precise, 1 in 241 cases, they actually collect the virus isolate and then they sequence the genome. The genomes, which is about 30,000 base pair, they are uploaded onto this database called GISAID. I'll keep talking about GISAID, so I'm just trying to set the context here. So GISAID actually is an influenza database, which has become the default database for all the SARS-CoV-2 sequences which are being uploaded. And you can see at the moment, at the bottom of the slide, I don't know if you can see it, there are around 80,000, just over 80,000 sequences from across the world. And that's a very exponential growth rate, as you would expect for a pandemic which is still ravaging. Right. So my laptop screen is having a lot of different subscreens inside it and preventing me from pointing. So I might be looking up. So pardon me if I don't make eye contact for a while. So let's try and understand how this virus evolves. Okay, so this is a schematic. So I've shown it like a DNA, but it's actually a single-stranded RNA virus. Most certainly came from bats, possibly through an intermediate host. And this aspect of the environmental biology is still being worked out. And what you see here in dark blue arrows, they're not actually one person giving this to another person. So the schematic, essentially, every symbol of a human being is actually a number of people. And then the arrows here in blue, they represent the volume of transmissions. So the virus is basically going from a bunch of people to another bunch of people to another bunch of people. And once in a while, when it moves across, it can accumulate, it can encounter a naturally occurring mutation, okay? And as it continues, it can also accumulate other mutations as it goes along. And so let's take some time to understand this, because this will quickly get complex. So the dark blue represents the volume of transmissions, and the nucleic acid symbols, where you can see the different little colors I've introduced, they are representing a particular mutation. It might be a substitution, a deletion, whatever. It doesn't matter at the moment. And what you find is that at some point you had a person who was identified as SARS-CoV-2, and they managed to get the virus isolated from this patient and sequenced, and they uploaded it on GISAID or GeneBank. And that is what we all agree as the nominal reference, okay? So we've all agreed as a convention that that's what we call as Wuhan Hu1, HU1, okay? Right, so the first sequence which was sequenced, isolate which was sequenced, and that's the one with the orange and pink mutation here. It may or may not be the original strain or isolate that first infected the humans. Okay, so this is when we first picked it up, and it might have appeared sometime previously. And that's what I'm representing here as gray. Now, it is possible that we might actually sequence and isolate at some point, say now, and this is what I'm representing by the green one, and that's in the bottom of the slide. So, we are doing it now at the time point T is somewhere now here. What you may find is this is actually phylogenetically closer to the one that jumped originally from, say, bats to humans, okay? And you can work this out. You can figure out that actually, logically, this might have come earlier than what we all currently think is the first one, which is the Wuhan Hu one here. Now, notice that from Wuhan, things have evolved, and we now have numerous circulating clusters, which is shown on the right-hand side here. And when you are doing this, in a bioinformatic manner, so for those of you who are from BITS, if you are a bioscience and computer science double major, dual degree, so this is your ideal sweet spot, right? So you're using machine learning and computer science to identify the cluster consensus, because this is an RNA virus which will exist as a quasi-species. So from your genome sequencing output, you are identifying the consensus, which is represented here by this terminal as a schematic. So you have all these various clusters which are circulating in different parts of the world. Now notice 4 things. The most virulent strain, meaning I'm using the word strain here to really mean isolate or a clade if you want to be pedantic virologically, but I'm going to use the word strain just to keep it simple, okay? The reason is twofold. One is the word strain is being used or misused by the media quite extensively. And also GISAID itself tends to use the isolates to, they use the word strain when they actually mean isolates. And nextstrain.org also uses the same terminology. So I'm just going to stick with strain at the moment. So the most virulent strain need not be the most circulating strains. And in fact, because of quasi-species theory, you can almost be confident that the most circulating one is unlikely to be the most virulent one. Okay, we can come to that later if this is of interest. Neither of these are the first strain that we all have nominally agreed to be the reference one, the nominal reference. And these 3 need not be the one that is closest to origin, which might still be circulating somewhere, which you may find out tomorrow is actually, from an evolutionary point of view, much closer. Okay? So this is the abstraction. Now, so this here says that, the 2 currently circulating strains, which what I'm showing here in, orange, pink, purple or orange, pink, brown here, need not be, the one which is the most virulent here, and that need not be the one which is the closest. Right, now let's move on. So what is the probability that we might find something older than the Wuhan nominal reference? Okay, so the genetic code of the virus, actually China was pretty prompt in releasing the sequence of the virus. Okay, so around 5th of January. And this is what spurred the entire vaccine discovery, because a lot of the researchers around the world were using their existing platform and slotting in the SARS-CoV-2 sequence to quickly develop the vaccine candidates, including the 2 vaccines which I've been involved with in terms of preclinical research. Now, my colleague at UCL, Francis Ballau, was saying that based on phylogenetic estimates, you could say that the pandemic probably started sometime around between 6th of October and 11th of December, although the first sequence was released a bit later on the 5th of January, okay? And 2 very curious news items emerging from different parts of France seems to give credit to this theory. First is that there was a patient in a hospital near Paris on 27th of December. This was before the 5th of January. And this basically shows that the virus could have arrived in Europe, much earlier than we believe, okay? And they actually tested and retested, and it did come back positive for COVID-19. And then there was another case in the Albert Schweitzer hospital in Eastern France. And what they found through, medical imaging is that this actually, by looking back retrospectively at chest scans, they found that even as early as November the 1st, it's possible that we might have been seeing COVID-19. Once again, this goes back to the October 6th date, okay? So you will find more and more of these sort of news. So don't be alarmed. This is what we intend to discover. Now, why is all this important? Why is it important to study how this virus is evolving? Should we be alarmed that the virus is mutating? Now, this is an RNA virus, so I'm not personally too bothered that it's mutating. That's what one would expect. But let's think about vaccines and therapies, right? So many of the vaccines and therapies are developed in the discovery phase by taking a sequence which is made available, and then they slot it into, say, a Vaccinia platform, or in the case of Oxford, they've used their ChAdOx, which is the chimpanzee adenoviral vector platform. So they tend to use the genome sequences which pertain to typically one of the earlier isolates that we encountered. Second reason is that animal models where you have to test these vaccines and therapies before you can go to the phased clinical trials and ultimately human use authorization. These animal models, whether you are looking at ferrets, which is my specialty, or whether you are looking at non-human primates or other models, they often tend to be developed with the first available virus isolate. So Shrikar, you introduced me saying that, we were the first outside of China. Now, we've done a lot in our team that's probably more exciting than being the first outside of China. That happened to be good fortune that, the whole of the world was waiting for an imported case, because when you have the genome sequence made available by China in early January, to actually develop an infectious clone, which is like a reverse genetics. So basically you're trying to recreate the virus, takes months. So it's a lot easier to just wait for, the virus to arrive in the form of an imported case, and then to be able to isolate it and grow up the stocks. I'm not trying to say that that research is not important or hard. It is, when you are dealing with an unknown virus, to be able to do that. And we are proud of what we achieved, but that's just one of the things we did in the beginning to initiate the preclinical research in the world. And that was shared across. So my colleagues at the Victorian Infectious Disease Reference Lab isolated the virus because it's a human lab. So we are animal health side. So the virus then came to us, and it also went to Public Health England. And as you said, we were able to grow up the stocks in large quantities and initiate. Notice the suffix of all these, right? So we were using VICO1, which is Victoria 01, Public Health England, which I used to work for before I moved to CSIRO in Australia. They also use the same. The American labs tend to use Washington 1. Europeans tend to use the Bavarian BavPat1. So there's a reason why all this is 1-1, right? Because everyone is twiddling their thumbs and waiting for the case to arrive. So you get the first virus isolate and you run with it. So you develop your animal model. But at some stage, I felt that this diagram started to bother me. That I felt, okay, so we took something which is as close as possible to this one, but then with time t progressing, you might actually be ending up with something like this. And how representative is this one of what's going on here or what's going on here? So that's the question. So, and this is really important because when you are looking at an outbreak response, you are looking at two different things. One is you are looking at vaccines. So you want to prevent the virus from spreading. And bulk of that strategy is through social distancing, lockdowns, and vaccines. And the other one is when it invariably spreads, you want to make people feel better. So you are looking at therapeutics, and therapeutics are typically drugs or therapies like monoclonal antibodies or polyclonal antibodies and so on. So when you are looking at vaccines, you want to choose the most prevalent circulating strain and ensure that your vaccines are working against them, okay? Whereas when you are looking at therapies, you might want to pick the most virulent strain and make sure that the therapies which are being discovered or repurposed, that they are able to handle the most virulent form of the disease. Why can't you use the most virulent for vaccine? Because by doing that, you might artificially increase the threshold of evaluation and thereby reject perfectly acceptable candidates, okay? So this was the context. And let me switch gears and I'll circle back to this, okay? So around that time, as I told you, we are going back in time. In late January, my lab got the Victoria 1 isolate, and then from which we grew large stocks. That was in early February. And we were also the first in the world to show that ferrets are susceptible to SARS-CoV-2. And this was the Nature piece, which was talking about how, mice to monkeys, different labs are rushing to see which animal model would work. Now, the reason I chose ferrets was because of the long history, of use of ferrets in respiratory illnesses such as influenza, and it seemed a logical choice. The other one we were toying with was pigs, mainly because we are a large animal facility, and we are actually the world's largest operational PC3, PC4 complex with the ability to do large animals under GLP. It was good that, in a way, looking back, that we didn't choose pigs because pigs are not a good model for SARS-CoV-2, so we might have hit a dead end. But as it turned out, ferrets are a nice intermediate model between mice on one end and non-human primates at the other end of the spectrum. The way it worked was, so, you collect typically a nasopharyngeal swab from an imported case in this case, and the Victorian Infectious Disease Reference Lab isolated it from this particular patient because it's a human case, and they sent it to us, and we propagated it on Vero E6, which is a type of cell culture. And then you make sure that you screen for mycoplasma and you do the next-gen sequencing, and you then have to confirm whether this isolate is representative of what's in circulation. And this was the bit I was talking about earlier. Once you do that, and it's the same isolate which also went to England, etc., and then by late February, pretty much different countries of the world started to get their own imported cases. So say the United States had Washington 1, and then that was then sent around to the North American labs. So this then initiated a number of studies. So you want to do susceptibility, dose, time course, vaccine efficacy studies. So what do we mean by this, right? So susceptibility is you first want to ensure that you can develop an animal model of the disease. So you try and find an animal which is vulnerable to SARS-CoV-2, in which you can test before it can be, tested in humans. Sometimes, as with Ebola and so on, it's unethical to test in humans if you have run out of cases and so on. So what you then do is use the FDA's animal rule, whereby you are testing for efficacy in animals and use that data to actually go straight to licensure. In this particular case, there's no dearth of human cases. So the animal models are really complementing the various preclinical evaluation going on in the form of human clinical trials. The dose studies are to try and work out what dose of the virus to give to the animal model. And then if you are evaluating a therapy or a vaccine, you want to make sure that you can get the dosing right, okay? The time course study is really where you try and understand the natural progression of the disease in particular animal models. When you have done all three, which we did by sort of mid-March, with funding from a body called CEPI, which stands for the Coalition for Epidemic Preparedness Innovation. So CEPI is basically the global body which is coordinating worldwide efforts in terms of the vaccines discovery and development. So following our success in establishing the ferret model and in completing the time course studies, we were given a grant to look at the vaccines, Oxford's vaccine and Inovio's vaccine in animal model. So just to give credit, while we were leading the pack, we were no means the only ones. So there's been a lot of research, and we're all, the correct way to say is not, standing on the shoulders of giants, but it's more like, I like to give the analogy of emperor penguins, going around in a circle, trying to hide behind each other and doing this giant circular dance. So every research group comes up with a new discovery. So I sit in these WHO animal model and other working groups. So we meet every week and we share information so that we don't have to reinvent the wheel. And there is extraordinary amount of information sharing at the moment going on. And these are some of the major research groups around the world which have used various isolates. Again, look at the suffix here, 1, 1, 1, 1, 2. So it's not uncommon to use one of the earliest isolates, right? And also, if you look at the, for those of you biologists, the tissue culture infectious dose 50, or PFU is plague forming unit. So it's basically a measure of how much virus you are giving to these ferrets and by what route. Typically, you give them intranasally, and it ranges around 10 power 5 to 10 power 6, thereabouts. So in summary, for ferret model, and the same, you can see that there are similar summaries, which I shall not discuss here, for other animal models. If you use that kind of 10 power 5 dose intranasally, you can find that ferrets are a good model of mild to moderate disease. You may or may not find clinical signs, but that's not critical for a vaccine evaluation. What's important is here, which is that the main model output is reduction in virus shedding as you measure in nasal washes, oral swabs, and rectal swabs. So, and these shedding usually happens between day 2 and day 9 after the infection. And when you are giving vaccines to the ferrets and then you are testing them, you want to make sure that the virus shedding is reduced. What do we mean by that? It's reduced in the amount of virus which is being shed, in the number of animals. If you are looking at it in terms of number of animals, you want to make sure that more number of animals are showing reduced shedding. And also the duration of shedding is less. So you want to make sure that the shedding is substantially less. Okay, so this is the general principle of animal models, which are now being published around the world. Many are available in preprints and some in published journal papers. There's actually a Nature review, which I'm one of the authors of, which is going to come out shortly, which summarizes all the animal models. And generally good animal models for COVID are ferrets, hamsters, and non-human primates, particularly rhesus monkeys. We don't work with them here, so we are generally focusing on the ferret model. Now, let's switch gears and move on to the vaccines, right? So now by March, we were ready to go and test the vaccines. And here are the vaccines which are funded by CEPI. Now, there are other vaccines which are Chinese vaccines, which I shall not cover in this talk. I'm only focusing on the global vaccines which are enjoying funding from CEPI. So the date you see in brackets are the date when CEPI announced funding. So I put it in the order in which they won CEPI funding. And these are the different vaccine candidates. I've color-coded it so you can see what CEPI is doing here is a basket of vaccines. It's hedging its bet, as it should, as a global body. It's not choosing any one particular type of vaccine. So you have protein subunit vaccines like the one in Queensland in Australia, or Novavax, which is going through clinical trials in Australia at the moment. And there's also the Clover, which is part of the Sichuan biopharmaceuticals. You have either replicating or non-replicating viral vector. So an example of the replicating viral vector is the Institut Pasteur, and the non-replicating viral vector is the Oxford vaccine, which I've been involved with. You also have live attenuated, like the Hong Kong, or a bunch of nucleic acid vaccines. So you have either the mRNA, which is Moderna or CureVac, or the DNA vaccine, which is Inovio. I'm also involved with the evaluation and preclinical testing of the Inovio vaccine. Now, there are no licensed nucleic acid vaccines for humans. So if the, say, for instance, Inovio ends up being licensed, it would be the first in the world, a DNA vaccine to be licensed for human use. The Oxford platform is a much different platform. So it's not a brand new one like the Inovio. So it's an established platform called ChAdOx, as I mentioned earlier. So ChAdOx stands for CH is chimpanzee, AD is adenoviral vector, and Ox is Oxford. So they use the ChAdOx platform, and now it's been licensed by AstraZeneca, so it has an AZ number. And this vaccine, as well as the Inovio vaccine, is given intramuscular, so as an injection. Inovio is also looking at intradermal. And one of the research questions I was interested in when I took this project on is to see whether we need 1 or 2 doses of the vaccine. And the second question is whether the Oxford vaccine, which is being given intramuscular, if it can be given intranasally, i.e. through the nose, like a nasal spray, like flu mist and so on in children, for instance, whether it will confer better protection than the intramuscular route. So these were particular research questions which I was interested in over and above what the developers were doing. So switching gears back, so we have been evaluating these vaccines since late March. We have finished the evaluation. We've shared the data with the developers. Due to various confidentiality reasons, I'm not in a position to share that just yet. So CSIRO is the National Science Agency of Australia. So we prefer to not put things as a preprint in bioRxiv and so on. We prefer to go through full peer review. And then when it's out, we can talk about it. So, so don't ask me on specific results, but the results have been shared with Oxford and Inovio and with CEPI, and they've gone to the regulators, and they are supporting the regulatory package at the moment because these vaccines are now either entering phase 3 or they are already in phase 3. So before human use, regulators will want to see evidence of how the efficacy and any safety signal in animals. And we are providing the ferret data, and there are other labs, particularly in North America, such as the Rocky Mountain labs, which are providing the non-human primate data. And so these go into the regulatory package to FDA, etc. Now, let's come back to the SARS-CoV-2 as a virus and mutation. So the rate of mutation for this virus, which is approximately about 15 to 20, so I put 18 there, that's not remarkable if you compare, for example, seasonal influenza, okay? The reason for that is that this RNA virus has a proofreading mechanism. For those of you biologists, it's an exoribonuclease in NSP14. And at any given time, if you look at isolates in circulation, they are no more than 10 mutations away from the ancestors that jumped into humans. And this is the diagram I showed you earlier. So at this stage, I do not believe we actually are looking at, strains like, I see people talk about Australian strain or Gujarat strain. I don't believe that is the case. So you are probably looking at a clonal population or clades at this stage. And I don't think that we are just yet looking at lineages. Having said that, there is one particular strain or clade, which is called the G strain, and it stands for switch from aspartate to glycine in the spike protein in the 614th amino acid. So that's why it's called D614G mutation. And it has been widely speculated as potentially being more pathogenic, potentially the transmissibility is higher. And there are also speculations that it could be resulting in higher fatality and so on. One of the things which is unmistakably clear at the moment is that the pandemic is the new strain. So if you go back in time, and bear with me for a minute. So this one here, which is the Wuhan Hu one, this is the old one, okay? So this is the D strain, D for Delhi, or aspartic acid. And what is currently in circulation here is the G strain, and G stands for the glycine, okay? So already what we see is that we have seen that the currently emerging dominant strain is the one which has the glycine rather than the aspartate. Whereas the earlier vaccines are all developed with this, and almost all the animal models in different leading laboratories around the world use this one. So one of the things I was keen to find out is whether this could be a problem for vaccines and indeed for therapies. Again, the paper is under peer review, so I can't talk about it too much, but I can say this much, that based on what we know at this time, we do not believe it's going to be a problem. So that's another interesting research which has been going on. So some of you might be familiar with the phylogenetic analysis, which is basically you're trying to track in the form of a tree, and this is an unrooted tree. So that's why it's shown in this way. And you start to see that very quickly an RNA virus, even with an exoribonuclease, will start to evolve and mutate. So you will see that there are different isolates which we have sequenced, which can be represented in this way. Now, bear in mind one thing, okay? And let's take a moment to go back here, and this is really important. So I talked about this. Only 1 in 250 odd confirmed cases are actually sequenced. So it's that Donald Rumsfeld unknown unknowns, right? So you don't know what you don't know. You only know what you have actually managed to sequence. The inherent question here is there a bias in the way the clinician or the reference laboratory decides which patient isolate needs to be sequenced? Is it that they are choosing particularly patients that they see are either more fatal or more experiencing a higher pathogenicity? That's an interesting research, but at the moment, let's simplify and say that there is no bias, okay, and that people are choosing at random. Assuming they are, then what you find here would be a reasonably representative picture of what's going on in the world, okay? So it's really important to bear this in mind and not get carried away. So, and I've color coded this so you can begin to see that actually I was asked this question in one of the news channels in Australia. And so I was trying to show them that there is no pattern. So when we published this paper, and this was the first peer-reviewed paper that I'm aware of, probably the first peer-reviewed in the world, on how this virus was mutating. So at this stage, we only had 181 genome sequences, okay? And so we did the analysis on that. I remember when I first wrote the paper, it was only about 115 sequences. And we submitted it to Transboundary and Emerging Diseases Journal. And it came back with some comments from the reviewer, and we had to address it. And it was a rapid communication with a 2-week turnaround. So we got the comments back in 2 weeks. And by then, the 115 had grown to 181. So I had to redo all the calculations when we submitted it, okay? Now, look at this graph, and this is really interesting. So we were at that stage in the outbreak, okay? So now, if the reviewers' comments come back, I'm probably going to look at another 1,000 to 2,000 sequences having added, pretty much. So it's very interesting in terms of how this is evolving. So not content with the phylogenetic analysis. So I was showing here that Australian isolates are all over the place. At that stage, this whole thing was splitting into C1, C2, and C3 clades. And then you can see C5, C4, and C6. So there were 3 new clades emerging. Don't get carried away by this, okay? This will branch and branch and branch. So don't get alarmed by that. And if you see something complicated like that, that's what you expect an RNA virus to do. So what we did was we wanted to do something better than the phylogenetic analysis. And this BITSian audience is probably a very interesting one where I can actually talk about it without having to explain too much, because you've done your probability and statistics, you've done your biology courses in your first 2 years, so you can understand pretty much everything I tell you. So imagine you take, so remember this is an RNA, but when you are sequencing, so RNA is basically uses 4 letters of the alphabet. So it is A, U, C, and G. So, and it's about 30,000, roughly speaking, base pairs, okay? So the way I like to explain this is, imagine you have a piece of paper in which you have written a story or a novel, and the total character count is 30,000, okay? That's what it is. So it's not an A4 paper, it's more like an A0 paper. And so it's a big paper. So you have 30,000 characters, and it has A, U, C, or G. And that's your virus. And you are photocopying this thing in a giant photocopier. The only difference is every time you photocopy, you are photocopying the photocopy. So you're not using the original, you are using the previous version to make new copies. And that's how this virus spreads. And you have different photocopiers. So it's not just the one photocopier. So you're making different copies, and then different people are taking those copies and making more and more copies in different photocopies. There might be a smudge in the machine, and some machines might work differently. And sometimes it's a faithful replica, and sometimes it changes. Naturally, it picks up errors, and then errors accumulate over time. And that's what's happening here, okay? The only thing is, when you are actually sequencing it and reporting it, you actually report it not as AUCG, but you report it as ATCG, because you're doing the cDNA, okay? So it's basically a single-stranded genome, which looks like that, 5' to 3', if you remember your Sandhya Mitra. So this is towards the C3' end. And so this is actually called an or an octanucleotide motif. I've chosen this as an example. I've color-coded G, A, and C, and T. And think of it like you can chop it in twos, okay? So the first time you chop it, it's a G and a G. But you can also chop it as a G and an A. or you can chop it as an A and an A. So you can make tumors in this manner. And how many possible tumors can you make? Because you have 4 letters of the alphabet, so you have A, T, C, and G, you can make 4 power K, which is, in this K, your K equals 2, because it's a tumor. So you have 16 tumors, okay? 16 tumors. Now, again, if you think about it, if you have a sequence of length L, you will have L minus K plus 1 K-mers. So in this particular case, you will have 8 minus 2 plus 1. So you will have 7 tumors. What do? You plot this. You plot this as a probability function. So you go and count how many AAs have you got? In this case, only 1. So I put a 1. How many AGs have you got? You got 2. You got an AG here. You got an AG here. So you put 2. And so on and so forth. Where you have none, you put 0 or whatever. So this is a fingerprint. So if you connect all the dots here from 1 to 0 to 2 to 0, 0, 0, 0, 0 to 2, and so on and so forth, you get a fingerprint. And that is a unique signature. It's a tumor signature for this particular sequence. Okay, now you can increase the complexity. So if you do a 10-mer instead of a 2-mer, you will end up with 4 power 10. Now you can't do this with pen and paper anymore. You need to use bioinformatics softwares. So we worked with our high-performance computing team and my colleagues there, led by Dennis Barr. And what we did was to look at this and in an alignment-free way. So when you do this, you no longer need your rooted, unrooted phylogenetic tree. You are actually looking at a genomic signature of a piece of genome sequence, which is like this. And you try and plot it across the two, it's actually an n-dimensional plot, but you can do what we call a singular value decomposition and look at realign your axes so that you have the principal axis, component axis 1 versus principal component axis 2. And then your Wuhan Hu1 reference strain is right bang there in the middle here. So that's your black one. And then all these other sequences are all over the place. Now, in the box, inside the box on the top left, what you see is a comparison between where SARS-CoV-2 is here, and then you're looking at whether the SARS strains, SARS is SARS-1, they all cluster here. The MERS, the Middle Eastern Respiratory Syndrome virus, they all cluster there, and the SARS ones cluster here. That's reassuring. It will be problematic if they are all one on top, right? So they shouldn't be because they have unique genome signature. But then within that, we have zoomed this further here, and what you find is how these various isolates are behaving. And what you do is, the more complicated it gets, is we wanted to look at how the different isolates that are being used in different parts of the world. So you have the Wuhan Hu one here, and then you have the Washington one, and then you have the Australian ones, and so on. And what we found was that most of the isolates which are being used across the world for animal model development were indeed much close to each other and to the reference strain. So they were quite representative of what was at that point of time. And that was pretty reassuring, because what we were able to show is that the isolate we were using, and indeed the isolates which other people are using, are actually good choices. Okay, but then this is a very dynamic field. You have to keep doing this with time to make sure that you are still on track. And what did we find? What we found is that, these are a list of mutations in the particular isolate that we were using. And we ensured that this isn't giving us any significant headache in terms of the animal model development that we were using. And what you can do now is we've created this nice plot where you can actually see this in real time. where so this basically is a cluster of, tens of thousands of genomes, genome sequences uploaded on GSIRO, and you can see it in a nice single plot. And you can actually go and see where your favorite isolate is in relation to the Wuhan reference, as well as some other, you can compare the distance between two different ones. What does this tell you? If this is the black one, and you are looking at this one here where my cursor is pointing, if the distance between these two are less, then essentially it means it's very close. If on the other hand, the distance is huge, then it means it has evolved or mutated much more in reference to the reference strain here. Okay, so this one here has a greater distance to this one than this one. Okay, so this one is therefore closer than that one. That's what it means. Okay, now the next big question is this virus changing its pathogenicity? Now, this is a question you can't really answer. Now, ironically, even though you have 80,000 genome sequences, which are basically of this RNA virus, you don't have much information at all related to the patient metadata. So this gives you a word cloud, and the word cloud here, the word cloud here tells you that mostly, so the reference laboratory inputs some information, usually patient de-identified information, when it is made available to GSIRO. And it tells you what they know about the patient who got this virus. Mostly unknown or not provided. And that's not useful, right? And what we then did was we reached out to GSIRO and we said, hey, much of this data is meaningless because we are not able to make sense of all this, 80,000 odd genome sequences. So what we have done now is write a follow-on paper with GSIRO as co-authors, where we actually went and defined the global metadata set. So we actually said, this is the standard information that you need to be collecting anywhere in the world. You might be in Junjunu, you might be in San Diego, it doesn't matter. If you have a patient who's COVID positive, and if you have managed to isolate the virus and sequence the virus, remember that's typically 1 in 240. And if you are going to upload this information onto a global database, this is the patient metadata, patient de-identified metadata that needs to go in. So these are things like, comorbidity. Is the patient having diabetes or hypertension? Are they a smoker? Is it a younger patient, older patient, male, female? What do we know about them? And that indeed can provide you with very important information if you can correct for the baseline and to be able to look at, say, if I get the virus, I'm usually miserable because I get the man flu. The same thing goes to my wife and she's all dandy, right? So sometimes the same virus strain affects two people and they react differently. So for us to make sense of the host pathogen interaction, we need this background information. So this paper is actually under peer review. It's going to come out shortly and will probably help define the global standards in this area. So 3 things to remember. Bioinformatics, particularly these new methods which I showed you, like the K-mer analysis and the PCA, principal component analysis, can give you enormous insights in terms of what's happening with this RNA virus. It leverages machine learning and big data to make sense of the currently 80,000 sequences which are available. And for the best insight, what we need is still we require experimental evidence from laboratories like mine. You need to marry that with epidemiology and bioinformatics. And in the end of the day, don't forget that insights are as good as the data you put in, right? So rubbish in, rubbish out. So we need good clinical information to understand and address questions about this disease. So I'll pause here and thank BITSA and Ayush, Shrikar, and Priyanka for persevering with me and the opportunity to present to this audience, and to Tada as well. Thank you.